Our next speaker is Karen Guillemin. Karen is currently a professor at the University of Oregon. She has her roots in pathogenesis and then turned to a very interesting model system for both studying pathogens and, as it turns out, commensals, and learned a great deal of really interesting novel information about the biology of commensals in a gut-like system. She's going to talk to us about fish, and again, appreciate your being here, Karen.
Okay, thank you. Thanks so much for the invitation, and I'm very excited to tell you today about our work in a model vertebrate that's allowing us to explore these complex systems biology of host microbe interacting systems. And so this is an image of the kinds of visualization that we can do in our transparent model organism. So you're looking at the intestine of a zebrafish larva, and this is colonized with a fluorescent bacteria, and you can start to just get a sense of what it looks like inside there, what these communities look like. So we've been talking today about the importance of, for humans and human biology, of their microbial associates, just the numbers of microbial cells associated with humans, and the importance of these microbes in many functions of humans and energy metabolism, organ homeostasis, and immunity. And I think sort of the bottom line is that we need to really be thinking about humans as systems of interacting host and microbial cells. And in order to do so, I would argue that we need to have model systems where we can address questions of how these systems assemble and how they work. And so our model system that we're using is this model vertebrate, the zebrafish. And we think it's a useful model. I want to tell you just a little bit about it. So this is an actual adult zebrafish, but we're going to focus in on this internal organ of the gut, here showing the bacterial constituents of that organ in red with fluorescence in situ hybridization. And this model has a lot of experimental advantages. First of all, zebrafish are vertebrates, and they have guts and immune systems that are very similar to that of other mammals, including humans, and in particular, they have an adaptive as well as an innate immune system. For experimental purposes, they develop very rapidly and are transparent, and they allow us access to a functioning digestive tract which we can visualize. They're very genetically amenable, and then also, importantly, we've developed methods to manipulate their associated microbial communities. So we can make germ-free animals, and then we can reassociate them with different bacterial communities. And just to give you a little sense of the timing here, so this is just time post-fertilization of these in the time course of these animals, and you can see by 5 days post-fertilization, these animals have a fully functional gut. They're initially sterile as embryos, and then as they hatch out of their chorion, that's when you first get establishment of the gut microbiota. And also importantly, there's a staged development of their immune system. So in this early period, they're functioning with an innate immune system, and the maturation of the adaptive immune system happens later at 4 weeks of age, which allows us experimentally to separate these two types of immunity. And we're going to be focusing in today's talk on this earlier period. And so we're interested in studying host microbe systems, and the kinds of questions we focus on are on the assembly, the dynamics, and the evolution of these host microbe systems. And what our models allow us to do is to build up these systems and look at increasing complexity. Starting with the germ-free animal, we can then associate them with very simple communities, or we can allow them to be colonized with complex natural communities, and we can look at this whole spectrum of complexity. And in today's talk, I'm going to be focusing more on these simple systems and showing you what we can learn when we look even at these simple systems that, with just very defined microbial communities. And then actually in tomorrow's session, my colleague, Brendan Bohannon, will be talking more on the end of this spectrum in some of his comments he'll be making in his talk. And this work is actually funded now by a new center for systems biology funded through the NIGMS, which is devoted to these host microbe systems. And so this is there's a great team of investigators at the University of Oregon. We're the Microbial Ecology and Theory of Animals Center, or the Meta Center. And we just started as a center this fall, and we're very excited about the prospects of leveraging these model systems for understanding basic principles about host microbe systems. So first I want to tell you a little bit about what we're learning about the assembly of these microbial communities. And this is work that is really driven by my colleague, Raghu Parthasarathi, who's a biophysicist and also an expert in optics. And he and his two graduate students, Matt and Mike, have built this a really amazing microscope, a light sheet microscope that's allowing us to image these microbial communities in real time. And then there are a lot of other people involved, so graduate students, Zach Stevens, Jennifer Hampton, Adam Burns, and postdoctoral fellow Joshua Troll, who all have been contributing to this work. And this is just, principle, this light sheet imaging allows one to visualize in real time the fluorescence signals without inducing a lot of photo damage, because you're just illuminating your specimen with a sheet of light rather than a block of light. And it's allowing us to then visualize the gut of zebrafish that are colonized with fluorescent bacteria for long periods of time. So this is, I'm going to be showing you some images of movie of the digestive tract of the zebrafish that's outlined here in the dotted line. And this just gives you a little bit of sense that, so this is sort of somewhat similar to the stomach, a larger food-containing compartment of the gut than the vent or anus is out here. And I'll just show you what this looks like as it becomes colonized. Oh dear, these movies were showing. Okay, well, this might maybe, I have that some of the additional movies will show. But this is a way that allows us to visualize in real time the growth of these communities. And so what I want to tell you about is some simple experiments that we started to do to look at community assembly. And some of these are starting out very simply with communities where we're just actually looking at two strains, isogenic strains of dominant member of the zebrafish community, Aeromonas, that are differentially labeled with red and green fluorophores, and that's the only difference in their two genomes. And so we can colonize germ-free animals with these and then look at how the communities assemble. And starting out, we could think about Null hypothesis that if these two bacteria are going to have similar growth rates, one would think that if you were to perform the experiment of inoculating a second group at a certain delta T after the first group, you would expect growth to be similar in these for these two groups. And remember, these are isogenic strains. And so we can look first at the control of what happens if we just inoculate two strains at the same time. What you can see is colonization of the gut. These red and green speckles are actually colonies of bacteria that have grown up from that inoculant source. And if we quantify the fluorescence intensity along the organ of these red and green populations in replicate experiments, and so each of these is an individual intestine, you can see that the growth dynamics are really quite similar with these two populations. There's, sometimes the red is a little bit higher or the green is a little bit higher, but we see similar growth dynamics of the two populations inoculated at the same time. But then we can also start to perform the experiments where we now give one of those two populations a growth advantage, a time advantage, inoculate with a time interval. Here, just 3 hours time interval. And if you look, you might notice that there's a little bit more of the red here in this one, where a red population got inoculated. And I'm hoping this will play. Let's see. Okay, unfortunately not. So if this movie were playing, what would see is that in this case, there's a green population that got a head start, and it really dominates in the growth dynamics in this system. And so, fortunately, I have some quantification of those types of experiments where we've now included a time delay with different, combinations of either red or green first. And what I'm showing here on these graphs is the slope that one would predict for the growth of the second population based on the null hypothesis of equivalent growth. And what I think you can appreciate is that in fact, the second population has a much shallower slope. So there's a growth suppression in the case where we have this time difference. And importantly, this is happening before we've reached sort of any kind of plateau. So we can see in some of these experiments a plateau where we're starting to maybe reach the carrying capacity of that organ, but this growth delay is apparent even before that. And that's even more apparent at the 6 hour delay. So we're very interested in what could be causing these differences in growth dynamics, even with the same strain inoculated into the system, but with a growth delay. And so we're exploring the possibility that those first colonizers might have access to certain privileged niches within this organ. Also, whether there are changes in bacterial physiology upon colonization, perhaps in conjunction with changes in the host environment upon colonization. And one sort of example of that, and again, the movie's not showing, is that we can see that there's a lot of dynamics of immune cells in this organ. So this is showing a still from a movie of the neutrophils that line the gut of a zebrafish intestine, and we can visualize those cells with transgenic zebrafish expressing a fluorescent protein within that cell population. And we see that those cells are highly dynamic, and we know, as I'll be showing you in a little bit, that the presence of those cells in this organ is induced by the colonization of that organ by bacteria. So the early events of colonization are changing this environment and then changing, we think, ways in which later members might, the kind of environment that they're encountering. So now I want to switch to talk about dynamic interactions between members of this bacterial community and the host, and the kinds of experiments we're doing to explore these sorts of interactions in these simple systems. And this is work that has been largely pioneered by a new fellow in the lab, Anna Rolig, with important contributions from graduate student Yaya Yang, technician Erica Mitke, and postdoc Kat Milligan-Myrie. And so, oh good, at least one of the movies is showing. So this is showing a movie of the digestive tract where you can see individual bacteria that are moving here. And now this is a fluorescently labeled neutrophil in the system that's sort of standing watch there as these bacteria move around, and you can see this come up and taunt it a little bit, but this sort of this spatial relationship here between these cells. And Anna has gotten really interested in understanding some of the signaling that occurs that would induce the presence of these neutrophils in these systems. And so, as I mentioned, we know from earlier work that we've done that the presence of these neutrophils that we see in a normal conventionalized animal, and we can visualize these just with their myeloid peroxidase staining as these little brown cells within the intestine, is dependent on the presence of the microbiota, because absent in germ-free animals, and we can actually induce more of them if we expose the animals to a bacterial product like polysaccharide. So Anna was interested in understanding, well, are these the presence of these cells differentially induced by different residents of the bacteria of the zebrafish gut? And so what she did is took advantage of a large and growing collection of bacterial isolates that we have from the zebrafish gut, and she performed systematic monoassociations of fish. These are transgenic fish that have those neutrophils marked, and so she can, for each fish, she can record the number of GSP-positive neutrophils that are associated with the gut, and then she can also quantify the number of bacterial cells within that gut. And so this is just showing the quantification of the neutrophils. So each of these points is an individual fish that she's quantified. And you can see that there's a dramatic difference between our conventional and our germ-free, the blue and the green here. And then if you look at this range of different isolates, there's a real difference with some being very pro-inflammatory or inducing quite a large number of neutrophils, and others that have very little effect on the neutrophil number when they're associated with a germ-free animal. And she could go on and look at the relationship between the number of neutrophils induced in these systems and the number of bacteria that colonize that same organ, and do this on an animal-by-animal basis. And we imagined that there were several different possible outcomes of relationships that one might find. One possibility would be there's really no relationship between the number of neutrophils induced and the number of colonizing bacteria. Another type of relationship might be an inverse or negative correlation, where more bacteria would induce fewer neutrophils, or the opposite of a positive correlation of more bacteria inducing more neutrophils. And when she looked at those relationships, she found examples of all three types of relationships. So this is an Aeromonas strain that was actually very strongly induced neutrophil response, but there was no obvious relationship between the number of bacteria colonizing and the number of neutrophils. This example of a Shewanella species had a negative correlation, and Vibrio had a positive correlation. And so then Anna was interested in asking, well, what happens if she starts to build up simple communities from some of these kinds of representative members, and could one predict the host response to those communities just simply as the sum of the responses to the individual members? Or alternatively, is there more complexity to these systems so that there's really going to be some interactions between the members that give rise to the sum of the whole? Or are there sort of emergent properties that come out when we start to build up more complex systems than these monoassociations? So first, one thing that she did was to look at a combination of the Aeromonas and Vibrio strain. And here what I'm showing you for individual fish is the percentage of one or the other. So the germ-free fish are offered a 1-to-1 ratio of these two members. But what you can see is that in most of these fish, the Vibrio really dominated the community. And then she also asked, well, how does the presence of one or the other affect the fitness? And so one thing you can see is that if you look at how many Aeromonas shown on this lower graph, so the abundance of Aeromonas when they're either monoassociated or in the presence of Vibrio, the presence of Vibrio suppresses the growth of Aeromonas. Interestingly, the presence of Aeromonas enhances the fitness of Vibrio in this case, so that the number of Vibrio observed in the presence of Aeromonas is greater than in the monoassociation, or in cases where the Aeromonas was offered but did not succeed in colonizing. And then so we can look at the number of neutrophils, and what she noticed that in the combination there are more neutrophils present in this combined system, and she could look at that relationship more carefully. So here is shown just the number of neutrophils as a function of the colony-forming units of Vibrio in a Vibrio monoassociation, and then where Vibrio is offered Aeromonas, so there's none detected, and then also in the case where Vibrio and Aeromonas were detected. And this reflects that observation we made earlier, that you can see in general there are more Vibrio cells total when Aeromonas is present, and this relationship of increased number of neutrophils versus increased number of bacteria is maintained as a positive relationship. So, so that was sort of one interesting outcome. She saw a different picture when she combined Shewanella and Vibrio. So remember, Shewanella is one that had a negative correlation with the number of neutrophils and the bacteria. And when combined with Vibrio, it's a sort of similar story that Shewanella has a clear disadvantage in this competition. And in fact, the number of Shewanella present in an animal that co-colonizes Vibrio and Shewanella is suppressed relative to the number of when Shewanella is monoassociated. In contrast, Vibrio gains no advantage to having Shewanella there, so that's a difference compared to the Aeromonas situation. But then we also learned something very interesting when we looked at the relationship of the number of neutrophils versus the number of Vibrio colonizing. So if you look at Vibrio alone or with situations where there's no Shewanella detected, you see that very clear positive correlation that I've already shown you. But now, if we look at the cases where Shewanella is present, you see a real suppression of the number of neutrophils that are present. And if we look at on the other side of the number of neutrophils as a function of Shewanella, now you can see that this negative correlation is present. So this is telling us that in this diassociation with Shewanella and the Vibrio, now Shewanella's effect on the system is dominant, even though its proportional representation is really minor in the situation. So it's acting in a dominant way to suppress this neutrophil influx and inflammation. So we're starting to learn that there's some really interesting kinds of interactions that we can detect between members of the microbiota, and that those relationships impact the influence on the host. And the sum of those interactions could not simply be predicted by each individual member's effect alone, that there's emergent properties that come out of even these very simple systems. So finally, I wanted to tell you just a little bit about a new project that we're pursuing in this Meta Center, looking at the evolution of host-microbe systems. And this is a wonderful collaboration with my colleague, Bill Cresco at the University of Oregon, and is being led by Kat Milligan-Myrie, a joint postdoc, with important contributions from Zach Stevens, Mark Curry, Erica Mitke, and also not pictured here, another postdoc in Bill's lab, Clay Small. And this work stems from Bill's pioneering work in the model evolutionary system of the Threespine Stickleback, which he and others have built up into a really powerful system for looking at evolutionary population genetics. And it stems from the fact that we know a great deal about the population genetics and history of this fish, Threespine Stickleback, which if you explore the kind of genetic diversity present in these populations is quite similar to the genetic diversity in humans, and it has a sort of a similar length of history in which has been evolving, and some similarities to its evolutionary history in that it started out as an ancestral oceanic population, which then, with the recession of the glaciers, many independent populations were trapped in freshwater lakes. And those populations then have been isolated over time and have undergone interesting evolutionary trajectories. And there are some characteristic differences between these oceanic and freshwater populations. So, for example, the freshwater populations have lost a lot of their skeletal elements that would normally be important for protection against predators in the oceanic environment. And so there's a lot of differences, but importantly, one can perform crosses between these populations with in vitro fertilization, and one can actually map traits within F2 mapping families. And Bill and his colleague Eric Johnson have been doing this using RAD sequencing techniques to map specific traits within these populations to loci within the genome. And so Bill and I decided that it would be really interesting to use this kind of model system to start to explore the evolution of interactions of hosts with their microbiota. We're excited to be looking at these interacting host-microbe systems, and we're looking at these different dimensions of assembly, dynamics, and evolution. We're learning that in the assembly process, there's some evidence of the fact that history of exposures can influence microbiota assembly. So we're excited in exploring the mechanistic basis for this. We're learning that even from very simple systems that we're building up, there's evidence for microbe-microbe and microbe-host interactions that can really influence the dynamics and emergent properties of that system. And we're starting to explore how variation across different populations of host traits can influence the microbial composition, and we're excited to be moving forward those studies to actually start to identify genetic loci that contribute to community composition. So finally, I just want to thank all the people in the Meta Center who are contributing to this work. It's a really exciting team of people that working together. Also members of my own research lab, and funding from NIH, NIDDK, NIGMS, and the Birds Welcome Foundation. Thank you.
Thanks, Karen. Quick question, clarification? Karen, can I just ask, in the comparison of oceanic and freshwater stickleback intestinal microbiota, do you have, they enter the water system with their conventional animals, right? They have
so we actually make them germ-free initially, and then we put them into what we call system water. So they're reared in a facility that has recirculating water. So.
And so what does the microbiota picture of the water look like in comparison to their guts?
That's a great question. It's very different. Yeah. And Brendan, I think, might be touching on this point tomorrow as well. So,
okay, great. Because I have some more questions about that I can ask them.
Yeah.
Anything else for now?